Hello, good morning. It's a pleasure to be here with you today and to introduce you with the field of epigenetics, which is slightly different from what you've heard before, studying how genetic variants might influence your imaging phenotype, and doing something similar with epigenetics and trying to see whether we can also do EWA studies of imaging phenotypes. So this is probably the most complicated slide, trying to agree on a definition of what epigenetics is. Yeah? And so literally, if you try to ask what this means, it would mean outside conventional genetics. So that means forget about the SNPs and the other genetic variants that you've heard previously, and think about something else. And one early definition of epigenetics has been that it is a study of changes in gene expression that are heritable through cell division and that occur without a change in the sequence of DNA. While such a definition has not been very well received across all fields of science, because epigenetic studies is very fashionable and touching every field almost of research. So how do you define heritability, whether you are interested in more cellular aspects, like cancer researchers, or whether you're more interested in the individual, is very debatable. So the NIH came up with a definition that is more agreeable across fields, saying that for their project, epigenetics would mean to both heritable changes in gene activity and expression, and stable long-term alterations in the transcriptional potential of the cell that is not necessarily heritable. So it seems to be a very complicated definition, but you see that they touched at the definition of heritability, meaning cellular heritability or individual heritability. And they also mentioned changes in gene activity, meaning changes that would affect the chromosome, the genome itself, or the chromosomal landscape, but also post-transcriptional activities. So trying to be rather general and compliant to a broad field of researchers. Yeah. So very simplistically, we can say that epigenetics are the study of changes in phenotype without a change in genotype. And this is, epigenetics have been very useful in understanding, for example, how one cell, like a zygote, a cell in an embryo, the initial cell, can divide into give daughter cells that are so very different from the original stem cell and give a phenotype of more differentiated cells, like neurons, skin cells, or any different cells in the body, although they share the same genotypic information. And so I said that epigenetics has been controversial, very fashionable, because it has been shown to play a role in development, yeah, but also in cancer, immune disease, diabetes, and also for our own interest, mental health. Yeah. So one aspect of epigenetics, and we call it the epigenome, is that it is really reacting to the environmental factors, so reacting to the environment. Yeah, and this response is very dynamic, and it can be the environment defined as the intracellular environment, so the environment of inside the body, or external environment, so how where we live or how we live, the lifestyle can influence our genotype. So the epigenome integrates informations from the outside and the information in the genome to affect how we express those features of the phenotype. Yeah? So in a sense, so the epigenome enables the organism to adapt and evolve in a response to environmental stimuli. So I said that the epigenome is a very complex, epigenetics mechanisms are very complex. You have epigenetics mechanisms that affect the DNA structure, such as DNA methylation, or modification of DNA binding proteins like histones, there are also other epigenetic mechanisms that affect the RNA itself. So it can be very, very complex. For the sake of this lecture, we will focus on one form of epigenetic mechanism that is called DNA methylation. So then DNA methylation in mammals, it's usually occurred at a very specific position in the DNA at dinucleotides that are formed by cytosine and guanine in followed cell. So DNA methylation transforms a cytosine nucleotide into a methyl cytosine that is methylated, if I can move the cursor, on a 5 position. Yeah, okay.
It'll work if the mouse.
Okay, yes. Yeah, so it's a chemical modification of a very specific cytosine on a residue on the cytosine nucleotide. A specificity of DNA methylation is that it is stable, yes, but it is also reversible. It can be reversed by chemical modification of the DNA. And this is one particularity of epigenetics, including DNA methylation, as opposed to genetic variation that are extremely stable throughout the life of an individual. So what is the role of DNA methylation in the genome? So one thing that we should notice and be aware of is that DNA methylation is not regularly distributed throughout the genome. There is a non-random DNA methylation occurring at the genome, at genomic locations. So it is very common that there are clusters of CpG methylation sites, so CpG clusters at around promoter regions in the genome, and these are called CpG islands. And those sites of DNA methylation, highly dense and concentrated, they usually are non-methylated. And the fact that they are non-methylated allows gene expression and transcription. And if DNA methylation occurs at this site, that would just translate in inhibition, silencing of gene expression. Other regions of the genome, not close to promoters, can be also regulated by DNA methylation. And those regions can be highly methylated. And there are specific regions in the genomes that, when hypermethylated, can affect genomic stability, probably because on those areas are transposon, for example, or sequences that influence how stable the genome is. And so the consequence is that if those regions that are normally hypermethylated become demethylated, that results in genomic instability. And the consequences of both aspects, so in changing genomic instability or transcriptional activity via DNA methylation change, has repercussions for a whole range of diseases, and that are listed here. Now, I would like to highlight some commonalities and differences between GWAS and EWAS. So both, when you're doing both approaches, you're trying to actually screen 100,000 of variations of loci within the genome and trying to match that with a phenotype to see how genetic variants or epigenetic variants influence variability in your phenotype. While GWASs use SNPs, as you've heard before, to test association between disease and phenotype, for EWAS studies, we look for differences in DNA methylation at CpG sites associated with the disease and all the phenotype as for GWAS studies, but also there is the possibility to see how DNA methylation at those loci are related to certain environmental exposures or risk factors that might contribute to changes in DNA methylation. So this adds an additional level of analysis to EWOS studies. One thing and a difference between GWAS and EWOS is that the EWOS field is relatively new, and it takes also advantage of EWOS studies using same set of, or very similar set of tools to do genome-wide association studies. But there are some specificities of epigenetics, DNA methylation, that needs to be considered when performing EWOS or wanting to perform EWOS studies. And these are just a very simplified list of challenges that are associated with EWOS studies and that need to be taken into account before one even starts to design an EWOS study. So the first question is that because DNA methylation, like any epigenetic change, is very tissue specific. So you can have changes in DNA methylation in one tissue type that will not occur in another tissue. Yeah? So this is, again, a very, a notable difference between genetic variation and epigenetic variation. Epigenetic variation is very highly tissue-specific, or can be very highly tissue-specific. Yeah? And this is a problem for people wanting to do EWOS studies, and especially imaging genetics, because we would like to do that in living individuals. And it's therefore not always possible to use the brain as a tissue, as a source for EWOS studies. So one would want to consider other type of tissues, and of course blood is an ideal, very good candidate to be used as a surrogate for brain in those studies. Yeah. But one needs to be aware that epigenetic changes that are measured in blood might not necessarily reflect epigenetic changes that occur in the brain. Yes. And something very related to this, taking blood as or other tissues as source of DNA methylation, is that the cellular heterogeneity that occur in tissues. So in blood, you can have something like 50 different cell types in blood, and all those cell types might have different DNA methylation patterns that might influence your association studies. So this cellular heterogeneity needs to be taken into account when doing EWOS studies, so controlling for cell type diversities in such studies. I will touch also later in the talk, discuss the problem of sample size and power that are associated with EWOS studies, and also trying to touch about the problem with doing EWOS studies that is really different with GWAS studies, is that it's difficult with EWOS study to see whether your DNA methylation is a cause or the consequence of your change in phenotype. Now first, just a very schematic view of EWOS workflow. So what you need to do is obviously find your sample. And this is difficult enough when you do a GWAS, but when you do an EWOS, it's even more complicated, because you have to think about, okay, what type of tissue will I be using? And this really depends on the type of study that you want to do. Most EWOS studies use blood as a source of tissue for their genetic association analysis. Others use saliva. But blood has the advantage that the amount of DNA that can be extracted to measure DNA methylation is higher, and it's easy to access. So once there is, you have your tissue, you have to process the tissue in a form that you can really measure DNA methylation, and I will just talk to you through that very briefly. And then you measure epigenome-wide variation, like GWAS, using array technology to measure changes in methylation levels. And then, of course, the difficult thing is to analyze and interpret your results, but that's another part, yes. Just to mention shortly, the effect of tissue selection when doing your EWOS studies, and I will focus here, and in whether blood can be used as a good predictor for DNA methylation in brain. This has been studied, for example, in this publication, where they use about 120 subjects where they had blood and DNA extracted from blood and DNA extracted from brain, obviously post-mortem. And they tried to measure DNA methylation in both tissue and measure to which extent DNA methylation co-varied between blood and brain. And this is represented here in this graph. You can see that for most CpG sites, the DNA methylation variation measured in blood was not very well predictive of the variation observed in brain. So only a small percentage of CpG sites showed also high covariation in blood. Yes. But still, there existed a certain subset of CpG sites in the genome where there was a good covariation between blood and DNA methylation and brain. Yes. So that shows that it is possible to use blood as a source of tissue for EWOS studies, but this will, you have to take, to be aware that this might give you only limited information regarding the underlying biological process or pathological process. Yeah. But in some cases, it will also be very useful in identifying biomarkers of phenotypes that are really relevant for the brain by doing that. And some studies have been designed to test and to prove this. And this is what you see here is one of the first EWOS studies looking for association of the DNA methylations and Alzheimer's. And for that, they used post-mortem brain tissue for doing their EWOS studies. And you can see this is a Manhattan plot, the same type of plot that you find by using GWAS studies. And those they revealed hits, so CpG sites that were significantly associated with Alzheimer's disease, using brain as a source for EWOS studies. So that's fine, and they were happy. But as I said, it's difficult to obtain brain. If you want to use brain as a source of tissue, if you want to have information about the living brain, and you want to use this in imaging genetic studies. So the next step is then to see whether using blood DNA methylation that can be informative for psychiatric phenotype. The one they used here was schizophrenia. So this is a schizophrenia EWOS done using blood as a source for DNA methylation. And you can see that they have found some loci that attain genome-wide significance for association study. And the authors were only partially happy, and I can explain why if we have time later. But this shows that this really demonstrates that blood can be used as a source for EWOS studies in relation to psychiatry. Yes. So the next step was to see whether really blood could be used now to predict an imaging phenotype and predict behavior. So this is work by Barbara Ruggieri in our lab, and she used blood to see whether DNA methylation in blood could predict alcohol use disorders. So she first looked whether she could find differences in DNA methylation that discriminates between twins that were discordant for alcohol disorders, and she identified locus in the genome here that was differentially methylated in twins that abused alcohol and those that did not abuse alcohol. And then she went back further and she showed that this DNA methylation, or this change in DNA methylation, really could be found even before here adolescents were really exposed to alcohol drinking. So precedes the alcohol exposure and could predict the brain activity. Here is an fMRI in a particular brain area that is relevant for addiction. Furthermore, so these changes in DNA methylation and brain activation at age 14 could really predict changes in behavior later, so escalation of alcohol drinking and increased impulsiveness. Yeah. So that really showed that using a blood DNA methylation, we might be able to predict changes in the brain and changes later of behavior. So this finding encouraged us to expand those studies, and now to try to do in conjunction with Enigma, creating an Enigma epigenetics working groups where our aim is to find DNA methylation markers that affect and associate with the brain, like doing or expanding the GWAS studies that are being done in Enigma. Now I would like to give you some details about steps that are helpful in doing EWASs if you want to apply that to your own sample. And about sample preparation, so you have DNA extracted from the tissue of your choice that could be methylated at some loci. So how to measure DNA methylation? So the first step is to do a chemical change that is called bisulfite conversion. So converting non-methylated cytosine into uracil and then thymidine. Yeah. So this is done with this step. So what is shown here is that non-methylated cytosine will be converted into uracil, while methylated cytosine will stay and not be converted to uracil. And then the next step would be to do a polymerase chain reaction to amplify the DNA, and this amplification step will then convert the uracil that is contained normally in RNA molecule into thymidine. Yeah. While the methylated cytosine will stay as a cytosine, non-methylated in this case. So the procedure basically transforms non-methylated cytosine into thymidine. Yeah. It is good to know that those chemical reaction is very harsh. It depends on pH and temperature, and if it can result in DNA degradation, and that was concern early in DNA methylation studies, but now companies have improved their kits, commercial kits that now that have been tested better in terms of DNA yields and efficiency. The next step that is to do epigenome-wide DNA methylation profiling, and there are a whole range of epigenome-wide profiling, some that depends upon enzymatic reactions and then immunoprecipitation or PCR techniques. There is now also high-throughput sequencing for identifying DNA methylation profiles. Now, most people use still microarrays for this, and the microarrays mostly Infinium methylation array have evolved in their density of CpG sites on the array. Initially, 27,000 sites were represented on the arrays. Now, 450,000 CpGs are on the array, covering most reference sequence genes, but even there are now a new methylation array that can cover even more the genome in terms of DNA methylation. And what you get after hybridizing those arrays with your sample for DNA methylation is get those type of profiles that are a signal of unmethylated and methylated signal, and you have to compute a value that quantify the methylation. And this value is computed by taking the ratio between intensity of the signal of your unmethylated probe divided by the total number of probes, methylated and unmethylated probes, and that gives a beta value. Yes, and this is, you see here the distribution of beta value in a sample. It usually ranges from 0 to 1, and you have a distribution such that most sites are either non-methylated or methylated with a beta value of 1, and then you have some sites with intermediate levels of methylation. So as opposed to DNA sequence variation, where you have either a mutation, that is an allele that is there, as homozygotes, heterozygotes, or homozygotes for the minor allele. Here you have DNA methylation occurs via a range of values between 0 and 1. DNA methylation also can occur, occurs as individual CPG sites, individual loci, and one can use those loci as like a single nucleotide polymorphism in doing those association studies, like in a GWAS. But because DNA methylation occurs very often at what I said before, CPG islands, where you have clusters of CPG sites that are co-methylated together. So taking into account those adjacent sites that are methylated or demethylated together to identify regions instead of individual loci that are differentially methylated in the genome. And you can see here a comparison between a normal cell and a cancer cell DNA methylation profile in those two groups. Identify here a region that really shows that the cancer cells here are more methylated in these particular regions. So again, by doing EWAS studies, you can do the EWAS studies taking single positions as input for your association studies, but also considering strategies to trying to detect regions of methylation that are, in principle, more biologically meaningful. And then there are QC steps that are necessary, like in GWAS studies, when doing EWAS studies. One thing, and normalizations and back corrections for removing biological variations. Biological variations that are caused by batch effects or technical factors. And this graph here shows how this correction is very important. So this is what you see, inflation that is caused by non-correcting the intensity values, and inflation that has been almost normalized and disappears after correcting or performing normalization and batch correction for before your association studies. Also, two was about cellular heterogeneity and their consequences for your result. This is due to differential expression of DNA methylation within cell types. So if you have DNA methylation that differ in only one cell type between cases and controls, the DNA methylation differences that you identify between groups might be due to appearance in only a small percentage of cells. And also it could be due, the association difference that you measure, to the fact that your cases and controls will not have the same proportions of blood cells, types. And those affect, so cellular heterogeneity affect EWAS studies, and this cellular heterogeneity needs to be controlled for in EWAS studies. So there are different approaches for correcting for cellular heterogeneity. One is to just count the proportion of each cell type in the blood, so doing that, and then correcting for that in regression analysis. The other option that is simpler is to use reference information. Because cell types in blood are known and their proportions are known and have been computed already, and using those information to estimate cell proportion in your own sample. And for blood, it's easy because reference exist, but in other tissues, reference estimates of DNA methylation per cell type is not available. So there are methods that are reference-free to correct for cellular heterogeneity. Yes. And here is just for your information of a list of packages that are very helpful for processing and analyzing your data for DNA methylation studies and EWASs. They are all free and downloadable in the database and very, very helpful. And you can see from the list that that you can use here. And just this last slide to say that this is a new approach and really in need of improvement because many assumptions that are used in the analysis of DNA methylation are violated. And these are just some examples listed here. For example, DNA methylation, the variance in DNA methylation is really depending on the mean DNA methylation. Yeah? This is something that is called heteroscedasticity. Also, the CpG site density, as I said before, is not a constant across the genome, and usually DNA methylation is associated with high CpG density. So all that affect statistical analyses that are used currently with DNA methylations. Yeah. About sample size and power, this is something that is difficult to calculate for EWOS studies because, again, you have a great variation of DNA methylation in the genome and a great variation of DNA methylation depending on the cell type that you are studying. And so far, there has been very little information about frequency of DNA methylation changes and their effect size in common diseases. Yes, and so there are really no real precedent, like for GWAS studies, as to what to expect for effect size. But it is very likely to be varying depending on your genomic context. And also, as to DNA methylation being a cause or consequence of the phenotype that is measured, study design can help in answering this question. And one study design that is really helpful in investigating causality in terms of DNA methylation being the cause for a disease is the longitudinal study design, where you really collect DNA methylation and you phenotype in a longitudinal way in the same individuals. And this can really tell whether methylation is causing the change or not. Another way is to also to integrate genetic information, so GWAS information into your EWAS, because one thing with GWAS is that you can really say that your genotype predict your phenotype. Your change in genotype is causing the phenotype, so something that you cannot extrapolate to DNA methylation. So trying to do integrate a genotype that can affect your DNA methylation status. These are called methylQTL, and see whether those genetic variants also affect your phenotype and trying to generalize causality. My last slide, and this is an example of a study that used such a certain approach, combining GWAS study with EWAS study, and try to identify DNA methylation variant that mediate the risk of one disease, in that case, rheumatoid arthritis. And they use an approach that is highlighted here, and you can read a causal inference test, where they first look for association between DNA methylation and their phenotype, and then genotype and DNA methylation, so trying to find methyl QTLs, and then association of the genotype with the phenotype. So trying those different steps, combining genotypes and phenotypes to answer your questions. And finally, these are just some useful links for packages that will help you in your analysis. Yes. If you have any questions. I'm sorry for extending the time so long.
Thanks so much. Thanks so much.